Then you are giving here stain. Which stain you are giving here? Crystal violet or basic dye you are giving here. When you are adding this crystal violet, you will see that the color of the cell is bluish purple. In the second step, add here iodine, 0.5% iodine. This crystal violet will add with iodine and it will form crystal violet iodine complex. Crystal violet iodine complex. Okay. Then add alcohol to remove the stain, to remove the color. So gram-positive bacteria will retain the color because they have a thicker cell wall, so they will retain the color. Whereas gram-negative bacteria will lose their color. Why? Because they have thin cell wall, thin layer of peptidoglycan, and due to LPS layer, so they will lose the color. In the fourth step, you add another stain, that is safranin. After adding safranin, you will see that pinkish color cell you will get. So at the end of gram staining, you will get two color of cell. One is bluish purple, one is reddish color. Okay, the bluish purple cell are gram positive bacteria and the reddish cells are gram negative bacteria. Clear, Supriya? Supriya, clear to you?
Yes, ma'am.
Any more doubts, any one of
Yes,
you?
ma'am.
Any more doubts, any one of you? Any more doubts? Okay, today I will start conjugation, students. So our today's topic is conjugation. Okay, students. So let us see conjugation. My voice is audible to all of you now.
Yes, ma'am.
Meet sir. Meet sir. Leave it. Okay, so today's topic is conjugation. Okay. The first step we will see that suppose male is sterile. Male is sterile. So you are crossing F plus cell with F minus cell. At the end, you will get F plus cell. Okay. Let us see the diagram. So this is a bacterial cell. Okay. This is the nucleoid material. And this is plasmid material. This is male. This is F plus. And this is donor. Here 6 pili formation takes place. So this is 6 pili. And this one is conjugation tube. Conjugation tube. Okay. This is another bacterial cell. In this bacterial cell, only nucleoid is there, no plasmid is there. That's why this cell is F minus cell. Okay. In the next step, what will happen, students? Two bacterial cell. Okay. Now this plasmid will get transferred into this bacterial cell. And this is the nucleoid part. This is the nucleoid part of another bacteria. Okay. So within 1 minute, this transfer takes place. Okay. Within 1 minute, this transfer takes place. Again, they have the ability to form 6PD. Why? Because here this plasmid is present and this is the nucleoid body. Now we have started with F minus cell student, but as plasmid has been transferred, so this cell is becoming F plus cell. Okay, we have started with male donor, and this is actually female, and she is recipient. But due to this transfer of F plasmid, now it becomes male. Because male are F plus and female are F minus. So it is F plus, now it becomes male. So this is one type of conjugation student. So what we have seen here, we have seen that female has transformed into male. Female changed into male. We have seen here, female changed into male. This type of process is called sex reduction. Here sex reduction takes place, but genetic recombination is absent. Okay. Genetic recombination is absent. Any genetic recombination takes place between the nucleoid and the plasmid? No. But sex change takes place. This is one type of conjugation student. Another type I am showing it to you. This type is clear to you? This type is clear to all of you? Students? This is clear to all of you?
Yes, ma'am.
And the second type is again the second part. Suppose male is fertile. This is the second category. Better to write B. One cell is HFR, high frequency recombinant. You are crossing it with F minus cell. At the end, cell is also F minus. Now, see the phenomenon. This is the bacterial cell. Okay. And this is the nucleoid material. And this is the plasmid part. The plasmid, when it is associated with the nucleoid material, it is called, you know that, this is called epizome. This is called epizome. And this cell is called HFR, high frequency recombinant. Or it is called super male. Okay. So what will happen, student? Suppose this is bacterial cell and it is another bacterial cell. Like this. Okay. This is one bacterial part. This is the epizome part. Yellow is the epizome part. Okay. This is the bacterial nucleoid material. This is F minus, we know that. And it is HFR cell, we also know that. Now after 40 to 100 minutes, part of DNA is transferred. Part of DNA is transferred. You can see part of DNA. This one is yellow, plasmid part. And here also transfer of DNA takes place. So at the end, students, this is the bacterial cell. Okay, this is the bacterial cell. It is also F minus, but now this cell is HFR. We can say that these bacteria come from this bacteria actually, na. So this part is called exogenote. Exogenote means come from the outside bacteria, and this is the bacterial own nucleate material. So this is endogenote. So genetic combination takes place with their bacteria with another bacteria. So this condition is called partial zygote. Partial zygote, or better to say, it is called merozygote condition. Okay, merozygote condition. Now here, students, genetic recombination takes place. Genetic recombination takes place, but sex duction is absent here. Sex duction is absent. So two types of possibilities are there during conjugation. Clear with this?
Ma'am, partial zygote or?
Merozygote, partial zygote or merozygote. Okay. Clear with this? Clear with conjugation? Clear with conjugation, students? Our third genetic category is transduction. Okay. Transduction. So transduction means we are applying bacteriophage. This is a bacteriophage, student. Okay. This is the head portion. This is the neck portion. And this is the tail portion. Okay. Inside double-stranded DNA is present here. It is double-stranded DNA. And this outside coating is made up of protein. Okay. And this is the structure of bacteriophage. Okay. Now, this is a bacterial cell. Suppose you want to get infection in this bacterial cell. So firstly treat with this bacterial cell with lysozyme. Because when you will treat it with lysozyme, its cell wall will degrade. Okay, na? Peptidoglycan layer will be digested with this lysozyme. I've told you the function of lysozyme in previous lecture, student. And this peptidoglycan layer will be digested. Okay, there will be digested. This double-stranded DNA will enter into this bacterial cell. This is a bacterial cell. So this double-stranded DNA will come into this bacterial cell. Now the bacteria contain its own DNA, students. Suppose this is the bacterial own DNA. Okay, this is bacterial DNA. And this is viral DNA. This is viral DNA. So when infection takes place, what will happen, student? This is the bacterial DNA. This bacterial DNA combined with the viral DNA. Okay. This condition is called prophage or provirus. Now, this integrated virus cannot cause harm to the bacteria. Okay, it will not cause harm to the bacteria. So, this type of bacteria is called lysogenic bacteria. Lysogenic bacteria means virus is integrated. Okay, and these viruses are called temperate viruses. Temperate viruses. And this life cycle of virus is called lysogenic life cycle. Lysogenic life cycle. This is the lysogenic life cycle. Lysogenic life cycle is clear to all of you? Lysogenic life cycle is clear to all of you? Lysogenic life cycle, students?
Yes, ma'am.
Now, next one is lytic life cycle, student. Okay, so what will happen? The virus particle which was integrated there, that virus particle will come out. Okay, so viral DNA will come out. Viral DNA will come out. What type of excision, student? Asymmetrical excision. Okay, asymmetrical excision. So what will happen? Suppose this is the bacterial cell. This is bacterial DNA. Now this bacterial DNA merge with viral DNA. Change the color first. This is. Suppose this is viral DNA. Viral DNA I have shown with black, and this is bacterial DNA. So this DNA come out from that nucleic material. Okay. So what about the rest material, student? That it will draw with blue. This material is totally degraded. This viral particle, viral DNA, bacterial DNA comes out. Now they will use the machinery of bacteria. They are degrading the nuclear material. How they have degraded the nuclear material? With the help of nuclease. Okay. This nuclease, with the help of this nuclease, they digest vDNA. vDNA is digested. Okay, they digest the bacteria of, they digest the DNA of bacteria. Yeah. Now this viral DNA from the different body parts, they will utilize the machinery of the host. Here, which is the host? Here, host is bacteria. Okay. So they will make different parts. So thus, they will make the head of the bacteria, and this is bacterial DNA, and this is viral DNA. This is the head of. They assemble their body part, means head and nucleic acid body parts assembled. Now this is the bacterial. This is the bacteria. These are the bacterial DNA. This is the viral DNA. It is the head part. Means assembling takes place. Okay, assembling takes place. Now what will happen? Their bacterial cell will cause lysis. This virus will cause lysis of bacterial cell. Now this phase is called transducing phase. Transducing phase. Why? Because here contains bacterial DNA as well as viral DNA.
Ma'am, which phase? Ma'am, which phase?
Bacterial DNA, this phase, transducing phase. That means when bacterial DNA and viral DNA has entered into the head of the patch, bacterial patch. Okay, and this will cause the lysis of the bacterial cell. This will cause the lysis of the bacterial cell. So this is called lytic cycle. This is called lytic cycle. Lytic means lysis of bacterial cell. That one lysogenic cycle. This one lytic cycle. Now suppose this bacteriophage again will cause infection to another bacteria. Suppose this is your bacteriophage that contain viral DNA as well as it contain bacterial DNA. Let me draw with black. This is bacterial DNA and viral DNA. Suppose this will cause infection to another bacteria. So with yellow, I am drawing the another bacteria. Okay. So these bacterial cell contain its own DNA. The blue I am drawing. This is the bacterial cell own DNA. And now again viral DNA will come and bacterial DNA of another bacteria. Try to understand the phenomena, student. These bacteria contain its own DNA. Plus it will get the bacterial DNA which this virus gets from their previous host cell. Okay. So the bacterial DNA which was of different host cell, that bacterial DNA is called exogenate. This blue I am drawing it. So this bacterial DNA is exogenate. This is bacterial own DNA called endogenate. Okay. And it also contains viral DNA. Black I am showing the viral DNA. Black I am showing viral DNA. So this bacterial DNA contains bacterial DNA of its own bacterial DNA from another bacteria which is called exogenous. Its bacterial DNA is called endogenous and the viral DNA. So now again this condition is called merozygotic condition. Merozygotic condition. Merozygote means partial zygote condition. Partial zygotic condition. So this is the lytic cycle of the virus. That is the lysogenic cycle, and this is the lytic cycle. Lytic cycle causes lysis of bacteria. That is lysis of host cell. With this, we have completed our bacteria part. So next, I will start cyanobacteria. Till this clear, student? Till this clear to all of you? Now, our next topic is student cyanobacteria. Cyanobacteria. But here, this cyanobacteria is actually blue-green algae. They are BGA, blue-green algae. It causes the class Cyanophyceae. Cyanophyceae. They are largest gram-negative bacteria. Largest gram-negative bacteria. Okay. And they are microalgae, smaller in size, and they are oxygenic bacteria. Oxygenic photosynthetic bacteria. Oxygenic means they will release oxygen. They will release oxygen upon photosynthesis. Okay. They are primitive anaerobes. They are primitive anaerobes. You know, student, whenever earth formation takes place, at that condition was anaerobic condition. So during the evolution of earth, first time oxygen come into the earth with the help of the cyanobacteria. The cyanobacteria, they perform photosynthesis. That is why they are primitive anaerobes, means they started. Yes, tell me.
Ma'am, after microalgae, what is written?
Microalgae, oxygenic photosynthetic bacteria. Oxygenic. Oxy means oxygenic. Okay. So when earth formation takes place, earlier condition was reducing. Reducing means no oxygen was there. Okay. The first time oxygen come into the earth with the help of this cyanobacteria. This cyanobacteria perform photosynthesis under anaerobic condition. And after performing photosynthesis, oxygen was released into the environment. And with this oxygen, first time the anaerobic condition of earth was converted into aerobic condition. So the cyanobacteria is huge important in the course of evolution of earth, in the course of evolution of life. Okay, more detail you will read when I will teach you chapter evolution. Okay, cyanobacteria has very much significant role. Okay. So first time oxygen come into the earth during the evolution of life or during the evolution of earth, that is with the help of this cyanobacteria or blue-green algae. So they are cosmopolitan students. Okay. Cosmopolitan, meaning of cosmopolitan means they are found everywhere. So if you see the occurrence, student, occurrence. They are primitive anaerobes. occurrence. They are cosmopolitan. Cosmopolitan means they are found everywhere. Where student? They are found on frozen lakes. They are found on hot water springs, hot water springs, and they come under association with eukaryote also. They are found in the association with eukaryotes also. Let us see two examples. Very important. This example is given in NCERT student. In NCERT, this example is given. The one eukaryotic example is cycas. Now, what type of root is present in cycas? They possess poraloid roots. This is also one question. Poraloid root is present in cycas. And which fungi, which blue-green algae is in association with cycas? That is Anabina. Anabina. Again, Azolla, one fern is there. Azolla is a fern. Okay, Azolla is a fern. The leaves of Azolla, they are in association. Again, same cyanobacteria, that is Anabina. Fern, the leaves of fern, example, Anabina. Now, what about their body structure, students? Their body structure, they are unicellular and they are multicellular. Example of unicellular is Anacystis. Example of multicellular, in multicellular you will get colonial. Example of colonial is microcystis. Microcystis. And example of filamentous. Example of filamentous. Now stop. Okay. Oscillatoria. Anabina. These are the example of filamentous blue-green algae. So let us see the diagram of a typical blue-green algae. Just we have seen diagram of typical bacteria now, students. Structure of bacteria we have seen previous lectures. So if you see the structure of cyanobacteria, structure of cyanobacteria. This is the outermost covering. This is mucilage. Mucilage. Then the second covering, students. This one. This is the second layer. This layer is cell wall. And the third layer. This one is the third layer. This is cell membrane. Okay. Now again here inward invagination of cell membrane. But this structure is not mesosome. Okay. This structure has another name called lamellazome. In bacteria it is called mesosome. In cyanobacteria it is called lamellazome. And with this lamellazome actually nucleoid material attaches. So this is the nucleoid material. Okay. Now, cytoplasm near the nucleoid material is called centroplasm. Cytoplasm near the nucleoid material is called centroplasm. Okay. And thylakoid layer you can see here. So see the thylakoid layer here. These are thylakoid layers. Okay. Presenting surrounding the nearby cell membrane. These are thylakoid membrane. Okay. These are thylakoid membrane. And in this thylakoid membrane, phycobilisome is present. Okay, in this thylakoid membrane like this, these are phycobilisome. Okay, these are phycobilisome. Phycobilisome is present. Okay, this is phycobilisome. Now, what is present in this phycobilisome, student? In this phycobilisome, three pigment is present. Phycoerythrin, which is red in color. Okay. Phycocyanin, blue in color. And the third one is allocyanin. Allocyanin. Okay. Light blue in color. Light blue in color. Okay. So this is the main pigment. Phycocyanin is the main pigment. Okay. These pigments all together they are called phycobilin. They are called phycobilin. Where they are present in phycobilisome. These phycobilisomes are present on the thylakoid membrane. Okay. In this thylakoid membrane, what pigments are present, student? The pigments which are present here, that is chlorophyll A. Chlorophyll A. Then the presence of carotenoids. Carotenoids. Okay. And then mixoxanthine. Mixoxanthine. Okay. Now this mixoxanthine and carotene together they are called carotenoids. They are called carotenoids. Okay. And the cytoplasm which are present here near this phycobilisome region, they are called chromoplasm. Okay. Centroplasm, chromoplasm. Now this is the structure of a typical cyanobacteria. Clear with this? Clear with this, students?
Yes, ma'am.
Now what are inclusion bodies present in cyanobacteria? Okay. So inclusion bodies, student, means storage material. I have already told you. Inclusion bodies means storage material. Like alpha granule, they stored mixopycin starch. Then beta granule, they store fat. Okay. Volutin granule, they store phosphate. Then cyanopycin granule, they store protein. Then polyhedral granule, they store enzyme. The name of the enzyme is Rubisco. Okay. This enzyme helps in carbon dioxide fixation. More about Rubisco we will read in the chapter photosynthesis. So these are the inclusion bodies of cyanobacteria. Clear with this, all of you?
Ma'am,
Clear with this?
below beta granule what is written?
Beta granule, they are responsible for the storage of fat. Volutin granules. Below beta granule, volutin granule. I have written volutin granule. Clear with this?
Yes, ma'am.
Clear with this, students? Now, next phenomena is complementary chromatic adaptation. Okay. Complementary chromatic adaptation. Now, what is this, student? Okay, this phenomena is called Gaidu-Cobb phenomena. Okay, Gaidu-Cobb phenomena. Now, it is the Gaidu-Cobb phenomena I'm talking about. Ability to change body colors. Ability to change body colors with respect to different wavelength of light. Now, what is this, student? See the phenomena. Suppose this is ocean. Okay. Now, in the surface, cyanobacteria are found. The surface cyanobacteria are blue in color. The cyanobacteria which are present in surface, they are blue in color. But in bottom, the cyanobacteria which is present, it is actually it is red in color. I'm not getting a red color, so I'm going it with green space. These are actually red in color. I didn't get a red color, so I'm drawing it with green. They are red in color. So they are absorbing blue wavelength light. Okay, they are red in color and they are blue in color. So why difference in color, student? In the surface, they are formed in blue color. In bottom region, they are formed red color. Okay, in the bottom region, there phycoerythrin is present. Okay, student, phycoerythrin is present. This phycoerythrin is red color. Okay, this phycoerythrin is red color. This phycoerythrin actually is present here. Try to understand the phenomena. This phycoerythrin is red color. Okay, but they absorb blue color. They absorb blue color light. They are red in color, but they absorb blue color light. Okay, so by absorbing blue color light, they appear red in color. Okay, and phycocyanin, they are blue in color. Allocyanin, they are blue in color. So here, due to more pigment of phycoerythrin, they are absorbing blue color light and they appear red in color. So when blue light reaches to the bacteria, the concentration of phycoerythrin increases, and due to this increase in phycoerythrin concentration, they appear red in color. And which are present in surface bacteria, they are phycocyanin, so they are blue in color. So this ability, the ability to change body color with respect to different wavelength of light, this concept was given by Gaidu-Cobb, and after its name, the phenomena is called Gaidu-Cobb phenomena. One organism but appearing two color due to different wavelength of light. Okay. Clear with this Gaidu-Cobb phenomena? Clear with this, all of you?
Yes, ma'am.
When blue light reaches to the bacteria, a phycoerythrin concentration increases. So this bacteria appears red in color. So they are absorbing more pigment. As a result, more light is absorbed. As a result, more photosynthesis. Photosynthesis also increases. Now, one important question for a NEET student, very, very important. Okay, why color of Red Sea is red? Color of Red Sea is, it is not like that red color water is there. No. Color of Red Sea is due to a cyanobacteria. The name of the cyanobacteria is Trichodesmium. Very important student, Trichodesmium. Due to this cyanobacteria, color of Red Sea is red in color. Okay. Now they help in nitrogen fixation, students. So how they are helping in nitrogen fixation? Let us see. They help in nitrogen fixation. They help in nitrogen fixation with the help of an enzyme called nitrogenous enzyme. The name of the enzyme is nitrogenous, and they fix nitrogen in anaerobic condition. Okay, oxygen can hamper this structure, nitrogenous. Okay, so here a structure is present, which is called heterocyst. This structure is called heterocyst. They have larger structure. They have thick wall. Cyst means always thick wall structure. And here phycobilins are absent. Okay, here phycobilins are absent, and it is pale yellow in color. Okay. See the structure of heterocyst. I'm showing you the structure of nostoc. Nostoc. These are the filamentous structure, student, of nostoc. In this filamentous structure, one larger structure is present. Okay. Now this larger structure is actually heterocyst. Okay. In this heterocyst, mucilage layer is absent. In this heterocyst, mucilage layer is absent because this heterocyst is an anaerobic device. Okay, it is an anaerobic device. So here mucilage layer is absent. It is a thick wall structure. Thick wall structure. And here ES1 is present. Photosystem 1 is present. Photosystem 2 is absent here. Why? Because this photosystem 2 is responsible for evolution of oxygen. That's why photosystem 2 is absent in case of this heterocyst, because they are responsible for oxygen evolution. Okay. Now this nitrogenous enzyme they are taking atmospheric nitrogen from environment, and they are converting this nitrogen to ammonia. And this ammonia is utilized in the formation of amino acid, and further they will form protein. Now few prokaryotes are there that possess nitrogenous enzyme. Nitrogenous. Clear with this, friends? Clear with this, all of you? Students?
Nitrogen to NH3, then
NH3 to amino acid. Okay, these ammonia are utilized by amino acid in the formation of amino acid. A means amino acid. Clear with this? Students? Next part is reproduction. So in reproduction, students, asexual reproduction, they go for binary fission. Then the second mode, asexual reproduction, they go for fragmentation. Fragmentation means filamentous cyanobacteria is there. This filament will break off into this. They'll produce fragment and they'll produce a new cyanobacteria. This is a filament. Second part is fragmentation. Third category of reproduction is heterocyst. Just now I have shown you in diagram now. This heterocyst will separate from main body. It will separate from main body and it will germinate. When it will get favorable condition, it will germinate. Okay. And after germination, new structure will form. A new structure will form. Another structure is echinite. Very important student. Echinite. What are echinite, student? Suppose these are the filamentous bacteria, filamentous cyanobacteria. Here they contain reserve food material. Huh? These are the region they contain reserve food material. They contain reserve food material. Okay. So these are actually thick-walled structure. Again, they are thick-walled structure. So when they form thick-walled structure, student, during unfavorable condition. Okay. So what are echinite, student? Cells containing reserve food are called echinite. They contain reserve food material. So these are what? These are actually reserve food. Echinite is thick-walled structure. Okay. Now under favorable condition, what will happen, students? Under favorable condition, this wall will break. Wall break, and it will form new cell will germinate. It will form new cell. So this is about echinite. One, binary fission, fragmentation, heterocyst, echinite, and last one more category is there, students. Last category is hormogonia. Last category is hormogonia. What is hormogonia, students? Suppose this is the filament. Again, now here mucilage is present. What is present? This is mucilage. This structure where mucilage is present, this structure is called Macarism. This structure is called Macarism. What will happen, student? These are dead cell. Macarism means these are dead cell, and this will disintegrate. Okay, and new filament will arise. That new filament are called hormogonia. Okay, dead cell will disintegrate. Dead cells will disintegrate, and new filaments are formed. These new filaments are called hormogonia. These are the mode of reproduction in cyanobacteria. With this, I am ending today's lecture, student. Clear to all of you, student? Mode of reproduction is clear to all of you, students? Mode of reproduction is clear to all of you.
Yes, ma'am.
Okay. With this, I am ending today's lecture, students. Okay. Till then, take care. Bye bye.